I think we are also seeing radiopharmaceuticals developed for a wider range of cancers. We do have therapies available for prostate cancer, neuroendocrine cancer, but we are seeing this expanding, and this will further expand over the coming years, which I think is a really exciting prospect. I think particularly the value that Crown brings is what we touched on before in terms of the value of the models that Crown have. In the radiopharmaceutical field, we can now offer our platforms of preclinical models to our clients to run radiopharmaceutical studies.
Welcome back. In part 1, Bryan Miller walked us through his journey from cancer research to radiopharmaceutical development and explained why this therapeutic class is capturing the attention of oncology researchers worldwide. Now we're shifting gears to explore how Crown Bioscience is scaling its preclinical capabilities to meet exploding demand, the strategic partnerships shaping the field, and the technological innovations that could define radiopharmaceutical development over the next 5 years. Let's dive back in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech.
Now, I'd like to touch upon a different part of the technology. There's a lot of innovation happening. Many are saying that we're entering into a new era for radiopharmaceutical innovation. What makes this moment or this season of time different? And how are you as a company positioned to help shape that evolution, this field as it scales?
Yeah, I completely agree. I think we are entering a new era for it. It's quite exciting what we're starting to see. And I think one of the things that is most exciting to me, I touched on it a little bit before, is the development of diagnostics. I think we are entering a new era, and it's certainly we're seeing increasing interest and increasing excitement about the potential of radiopharmaceuticals. And I think we are seeing increased interest in the theranostic use of radiopharmaceuticals. So this idea that you're essentially using the same material as both a diagnostic agent and a treatment agent. And I think there's huge potential in that. And that's something we are increasingly seeing coming through. And this has a lot of potential for patient stratification and rapid movement towards treatment of patients as well. I think we are also seeing radiopharmaceuticals developed for a wider range of cancers. We do have therapies available for prostate cancer, neuroendocrine cancer, but we are seeing this expanding, and this will further expand over the coming years, which I think is a really exciting prospect. I think particularly the value that Crown brings is what we touched on before in terms of the value of the models that Crown have. In the radiopharmaceutical field, we can now offer our platforms of preclinical models to our clients to run radiopharmaceutical studies. And I think that gives a lot of great opportunity to run studies with these very advanced preclinical models that we have available.
What kind of limits do these models have, and where do you see the development going? What kind of limitations are we able to overcome very shortly?
So I wouldn't suggest there's limitations as such. So as mentioned, we have a number of different platforms. So we can literally take a study from 2-dimensional cell lines to 3-dimensional organoids, into CDX in vivo, into PDX in vivo. I think that gives very strong workflow in terms of maturing a study over time. And by the time you're running your study in, say, a PDX model, you're working with a model that is going to recapitulate the heterogeneity of human tumors and will give you some very clinically relevant information in your study, which I think can give a very powerful data set.
Perhaps give us a definition once again of PDX and CDX. What does that mean?
For CDX, that's cell line-derived xenograft. That will be xenograft models, which will be a human cell line implanted into a mouse to form a tumor, either subcutaneously or orthotopically. For PDX, that's patient-derived xenograft. So we have set these models up from patient samples. They've never been in culture. They are grown in animals and then cryopreserved from the animal tumors. So they will retain a lot of the features of the original tumor. So they give you a more physiologically, clinically relevant output than a CDX would, where it's undergone selection of cell culture. PDX haven't undergone that, so they retain more features of the original tumor.
The power of using models is that you, as you highlighted before, you're faster, but the other purpose is also to have more predictive models to get closer to your targets, if I shall say. What are your strategies to achieve that?
I think I would say there's two main strategies that we use, and it depends whether you're at the stage of in vitro, if you're doing in vitro studies, or in vivo. In vitro, I think our organoid models are incredibly valuable for this. They form 3D structures. They begin to form the features that you would see in an organ. And I think that gives you a much more clinically relevant output from your in vitro study than you would get from, say, a traditional 2D cell culture model. In vivo, I think it's the value of the PDX models. As I say, they retain a lot of the features of the original tumor. And crucially, you don't have the uniformity that you would have, say, if you're growing a tumor from a cell line. By growing it from a tissue sample, your model is intrinsically heterogeneous, the way a tumor would be. So its response to your therapy will reflect much more what you will see in a clinical context.
I'm curious, Bryan, what I'm hearing is there's a lot going on in the space. There's a lot of innovation, a lot of changes, a lot of breakthroughs. What breakthroughs are you most excited about?
I think for me, it's the theranostics. I think that's something that I find very exciting, especially their applicability to hard-to-treat cancers. So something like, say, PDAC, pancreatic ductal adenocarcinoma, very, very hard to treat. Current treatment options are not that effective. The 5-year survival rate is still dreadful. And I think what we're going to see in the coming years is the development of radiopharmaceuticals with a theranostic application towards these types of very hard to treat cancers. And that's certainly something that I'm quite excited about. I can see a lot of potential in it. And I think you're actually starting to see more development in this space. And I think it's something that we will see advance over the coming periods.
And how do you balance in your role, on one hand, moving very fast, innovating very fast, but on the other hand, we're working in an industry where eventually everything will be regulated and already very early have these considerations about how are we going to regulate things? How are we going to make sure the patient will finally get what we are developing? How do you balance these two worlds?
Well, what I would say is I think robust quality standards are always absolutely crucial. At Crown, we're always very dedicated to delivering scientific studies of the highest possible quality. And in setting up our strategic collaboration with MDC, we have a partner who have similar values to us and are equally committed to the quality of their work. And by having these robust quality systems in place, this allows our teams to perform work rapidly to meet essential timelines while maintaining extremely high standards in the study. I don't think there's any conflict between high quality and research rapidly, because if you do a low quality study, that's what's going to set your timelines back. If you do things that isn't up to the necessary quality standard, then you're not going to get the answer you want. It will set your research program back. So I think it's crucial to work with partners who have very, very high standards of QC, very high standards of quality in everything they do. And that is really ingrained in the culture at Crown. I think our clients know the standards that we work to, and we're certainly good at delivering on timelines and delivering rapid research, but we'll do so with the highest possible quality standards as well.
Let's make this now practical. What advice would you give a smart biotech scientist that's curious about radiopharmaceutical development? Where should they start, or what should they do first? And what are the things that can wait until later?
I would say my suggestion would be to work with the right partners, because you need to develop a robust strategy for your study in terms of how you're going to target your tumor, how you're going to construct your therapeutic, what your preclinical models are going to be. So I think drawing on information from the best people in the industry is crucial. And I think that's a lot of the value that a CRO can bring. Certainly with Crown, we've got a lot of experience with helping clients in the preclinical area. And we can certainly advise you on the development of your study. We can help you design your study. We can help you choose appropriate models. As part of the Radiopharmaceutical collaboration, we can help you select isotopes and a labeling strategy for your test material as well. So I think partner with the right people can really help guide you through those early stages and make sure you have the correct strategy in place to deliver a successful study.
How do you make this partnership successful? Because entering into a partnership with the CRO or later on with the CDMO, sometimes people compare it with marriage. It's very similar. You have to select very well your partner. What is your approach there? Because I'd say not every company fits every CRO. There are differences, and it doesn't always make sense for a company to go to one because it's the best or whatever reason everybody recommends. Sometimes it makes sense to go to another, right?
I think people choose CROs on different criteria. Certainly at Crown, like I say, we do work with a very wide array of clients with different needs and different interests. And we can always tailor a study to the individual client. So one of the things that we always try to do is we do like to see ourselves as an extension of that client's lab. So we do work in close collaboration in the design of the study, and there's very open communication during the progress of the study as well. And I think the success is seen by the amount of repeat business that we get. And we do form these close collaborations with our clients because we are really dedicated to helping them achieve the objectives of their studies. And we've had the feedback time and again that they start to see us as part of their team. They almost see us as part of the research team, and they collaborate very closely with us in terms of delivering the study successfully.
Now, I would like to look forward into the future, since there's a lot of innovation going on. What is your picture? Where are we going to be in 5 years from now in this field?
Dynamic field at the moment. I think there is a lot of exciting progress that's already happening, and I think there is more to come. We're certainly seeing, rapid growth in the interest in radiopharmaceuticals. I expect this to continue in the coming years. What excites me is the prospect of increasing the number of cancer indications that have radiopharmaceuticals as a viable option for diagnosis or for therapy. It's particularly around hard-to-treat cancers. I still think we all know there's a core cancers that the treatment options are not really there. They're not where they need to be. And I think radiopharmaceuticals does offer us the prospect of really driving forward the development of new treatments in this area. So I think what I would anticipate is that we will see quite a big impact of radiopharmaceuticals in this treatment of these very problematic cancers in the years to come.
Before we wrap up, Bryan, what is the most important takeaway from our conversation?
I think probably the most important takeaway is just the level of excitement that there is in this area around radiopharmaceuticals and the potential that exists for radiopharmaceuticals in the coming years, particularly as pertains to oncology treatments. Of course, as we know, radiopharmaceuticals also have applications beyond oncology as well into different disease areas. And as I've spoken about, Crown is a really good partner for these types of studies, and we can really support you in this field. We've got a fantastic library of different models, both in vitro and in vivo, available for use in radiopharmaceuticals. And with our partnership with MDC, we are very well set. We have all the expertise in order to help you design and help you to deliver a highly successful radiopharmaceutical program.
Where can people get a hold of you and further have a conversation about your work?
People can contact me through LinkedIn, and I'd be delighted to give them any more information that they need on radiopharmaceutical programs at Crown, or indeed any other services that Crown offers.
Fantastic. I will leave the links in the show notes, Smart Biotech Scientist. And Bryan, thank you so much for coming on the show today.
Thank you, David.
And that wraps up our conversation with Bryan Miller. The radiopharmaceutical revolution is just getting started, and the preclinical strategies we discussed today could reshape how these therapies move from concept to clinic. If you're navigating the complexities of biotech development, whether in radiopharmaceuticals or beyond, I hope this conversation sparked some new ideas. If it did, take 30 seconds to leave a review on Apple Podcasts or wherever you are listening from. Your feedback helps us bring more of these insights to the biotech community. Thank you for tuning in. Until next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.